So, backing up, title, and I am speaking for my colleagues on a manuscript that is about to go in for review, but principally for Valerie de Crecy-Lagarde and Andrew Hansen at the University of Florida. But this was a large consortium of folks who were working on this project. 5 years ago, shortly after we published the minimal cell paper, I was contacted by Andrew Hansen from the University of Florida, and he asked me if I had ever thought about how many of our minimal cell genes might be protecting the cell from damage caused by sloppy metabolic enzymes. And I confessed to Andrew that I'd never even thought about that. So when we were listing the tasks that or functions that a cell has to perform, this isn't something I really considered. And so I went to visit the University of Florida, gave a talk. I met with Valerie and Andrew, and they explained to me in ways that, so we are taught biochemistry. You know, you had the Linenger textbook and you look at glycolysis and this is how it works. And they explained to me that in reality, most enzymes will make mistakes. You know, maybe one in 2,000 or one in 10,000 reactions, but you produce some metabolic byproducts. And sometimes these are dangerous, and especially for sloppy enzymes. So imagine you've got a sloppy enzyme that is supposed to, be doing this pathway of A to B to C to D, but on occasion it produces, X to Y, and the cell may require repair enzymes in order to prevent the formation of Z, which could be a dangerous product. And so I really had never thought about things this way. And this led to my team being involved in a project that is now, the paper is about to go in for review. There's a whole bunch of us from a bunch of different institutions, but fundamentally it was the folks at the University of Florida, Christopher Henry at Argonne National Labs and the University of Chicago, and Oliver Fine at the UC Davis West Coast Metabolomics Center. And so what I came to realize is that, um, we know about the Linenger pathways and how things work. But for instance, just looking at glycolysis, for instance, if you consider 4-phosphoerythroate by produced by glyceraldehyde 3,5-phosphate, glyceraldehyde 3-phosphate dehydrogenase, and the production of 2-phospho-L-lactate by pyruvate dehydrogenase. Both of these enzymes make metabolic byproducts that can be damaging to the overall process of glycolysis. And so it is essential that cells have enzymes that can degrade these, like 4-phosphoerythroate, 4-phosphoerythroate, or 2-phospholactate, that you have enzymes that can degrade these things. And so what Andrew and Valerie predict in this paper is that 14 to 17 genes perform the task of damage control. And most of these genes, they believe, are part of the 149 genes of generic or unknown function, and life would be impossible without these things. And so the process that they used to do this study, initially they do intensive bioinformatics to identify potential metabolic damage repair enzymes, and then they obtain standard genetic code versions of the candidate genes so that they could express these proteins in E. coli, and then use these in biochemical reactions where they would test the capacity of these synthetic proteins to react with a broad variety of substrates. And so based on this, they would come up with hypotheses about what the substrates are for a number of these different enzymes that they propose are involved in metabolic damage prevention. And the next step, we then, for the non-essential or quasi-essential genes, my team removed those candidates from JCVI SYN3A, and then we grew those and sent them to Oliver Fine's team at the Metabolome Center at UC Davis, where they compared the metabolomes of the gene deletion mutants with the standard JCVI 3A to look for metabolites present only in the mutant metabolome, and those may indicate the target of the protein. So, for instance, in the case of these gene deletions that we made, there were a series of 5 HAD hydrolases or phosphatases among the genes of generic function encoded by JCVI SYN3A. And so we were able to make knockouts for the protease 66, 77, 710, 710, we couldn't make that one. We were able to make the knockout for 728 and 907. And based on the metabolomics data and the biochemical studies done at the University of Florida, they were able to predict the likely substrates for 4 of these proteins. And this compares well with data from Mesoplasma florum. And so this has given us a way to really get a much clearer annotation for that group of genes. So this was one advance made. For some of the other proteins that the Florida group characterized, they were not able to identify substrates, but they are still convinced that these are still likely involved in metabolic damage control. Oops, let me keep going here. One enzyme, so essential gene JCVI SYN380 encodes both a nicotinate nucleotide adenyltransferase and a hydrolase. Now, Andrew and Valerie postulated that both of these would be essential. And so what they had us do is to knock out by making a his to alanine mutation in the hydrolase function, and they predicted that this mutation would result in, we wouldn't be able to make it, but in fact we could. So now they were aware that the hydrolase activity is non-essential, and they, in the paper, propose some of the substrates for this, which are different nucleosides. But this is another example of the kind of work that is going to be visible to all of you in what I think is a really terrific paper. What they come up with is at a conclusion, though, is that there is no doubt that the genes involved in preventing metabolic damage are essential for life, as evidenced by them making up about 3% of the genes encoded by this minimal cell. And with that, I'll stop and take any questions you guys may have. Stop sharing. Here we go. So,
Roland, how widely conserved are these genes usually?
Fairly widely, actually.
Yeah.
Mm-hmm. Now, the hydrolases are really common in a lot of bacteria, and it's unclear, the sequence diversity of our 6 hydrolases is fairly great, but they are widely conserved, certainly in mycoplasmas and in many other gram-positives. They're pretty close relatives.
Mm-hmm. Very interesting. Do you think this also exists in eukaryotes?
I'm sure it does. So
I would agree.
Andrew was explaining to me that there are a number of human metabolic sort of orphan diseases that are the result of damage to some of these metabolic damage enzymes. And so even if only 1 in 2,000 of the enzymatic reactions results in these sorts of problems, they can cause severe metabolic effects in people.
Yeah, thanks.
Mm-hmm.
Very interesting.
Yeah, Valerie de Crecy, I'm just confirming that a lot of these are very conserved.
Valerie, so great that you joined. I hope I did justice to your work.
Did justice, except my internet connection is so bad because I'm in the middle of the French countryside that I didn't know if I could speak, but yes, you did a good job.
Thank you very much. And I think you will be, when this paper comes out, which I hope won't be too long, you'll be excited with the results. And I think the really elegant work that Valerie and her team have done. And I think it will change how we think about cells a little bit. I had never thought about this before Valerie and Andrew educated me.